Okay, thanks, Adi. Itai, thank you for inviting me. And my name is Dudu. I have a confession. I do not study human viruses. I do not study the immunological response they elicit in human. But as Marcelo told me in the break, when you do a lecture, you should tell a story, right? So my name is Dudu, and I'm working on pandemics. Not only am I working on pandemics, I'm working not on the past pandemic, on a future pandemic. I'm working on an ongoing pandemic that is getting more and more severe every day. I'm working on the silent pandemic. I'm wondering if we can dim the light a bit because it's a, I don't know. Who should I speak to? Ah, yeah, thank you. Uri is, oh. More? I think so. No, sorry, but. Okay. I'm working on the silent pandemic, which is another name, it's hard to see it, but the global fight against antimicrobial resistance. Antimicrobial resistance is a true problem that causes to the death of hundreds of thousands to millions of people, depending on how you count it. And it's real and it's happening now. The reason it's silent that it's not one pathogen that's attacking us, it's just several different bacterial pathogens that attack us, and many of them we know how to treat, but many of them we are not able to treat. And one of the main causes, not the only, but one of the main causes of this pandemic is the ability of bacteria to transfer genes to each other horizontally. So, for example, I can imagine a state where I say, well, I like Adi's hair, and I have a stress that I don't have hair, and under this stress, I'm taking genes from Adi, and now I have a lashing blonde hair. So that sounds crazy, but bacteria can do that. And since hospitals have a very strong selection on antibiotics, it's enough that there will be one bacteria that has a resistance that it can transmit horizontally, and then it can create a real problem for the entire hospital. It's called the nosocomial infections, and we see it a lot. Okay, so that was the framing of the story. I will tell you today about how plasmids, these mobile elements that transfer from bacteria, are able to transfer so efficiently. And before I start talking about that, I want to talk about phages. So as many of you know, bacterial phages are viruses, interesting viruses that attack bacteria, and the bacteria have many different ways to combat these phages. Maybe the most known these days are CRISPR-Cas system, which is part of the immune system of bacteria in order to attack these viruses. Also very important are restriction modification systems that are also meant to attack exogenous DNA, and there are really growing number of defense mechanisms to defend bacteria from phages. This is from 2022 from the Sorek lab, which means it's grossly outdated, and they have like 10 times more system. It's really a very rapidly growing field. On the other hand, the bacteria themselves are not firing. They have their own way to protect themselves, and they develop anti-CRISPR or anti-restriction and other systems to fight back all those defense systems. So this is the premise, and now a brilliant student in my lab, Buria Samuel, asked, what about plasmids? So what about plasmids? As I mentioned, plasmids are DNA. They're typically circular DNA within the bacteria, and they tend to transfer from one bacteria to the other. So as a recipient bacteria, I receive a DNA that comes from a different source, and many of these systems will attack those, and we know that because we use restriction modification to cut plasmid all the time. We use CRISPR-Cas system to change plasmids. So how do plasmid transfer so efficiently, causing this silent pandemic and everything? So this is was a very good question, and what she did, she looked into 3 anti-defense mechanisms that are characterized, and we know for sure that are anti-defense because we wanted to have solid ground. She studied known anti-CRISPR, anti-restriction system, and anti-SOS, which I didn't mention before, but it's a way the bacteria protects itself when it feels under stress. And it's relevant for plasmid because plasmid mostly transfer in single-strand DNA form, and in that form, the SOS response of the cell is expressed and triggered. So Burya looked for these genes in plasmid, and she found quite a lot of these. They are marked here in red, so we were happy that they exist. But what was more curious, she says, I keep finding them next to the relaxase and the ORT. And in order to understand what it means, we need to go back to microbiology 101 and recall how plasmids conjugate. So the way plasmid conjugate, I like this one, it's a big plasmid, it will be fun to conjugate it. They have a conjugation system, so it's a kind of a tube or an injection mechanism that connects two bacterial cells. And then there's a location that's called ORT, which is the origin of transfer, not to be mistaken with the ORI or ORIV, which is the origin of replication. This is the origin of transfer. And then the relaxase nick the ORT, one of the strands, and then it binds to one of these strands and just take him to the cell into the recipient bacteria. Now, this is where Burya found all those genes. Next to the ORT, we know it's the leading region. It's called the leading region, the one that enters the recipient cell first, because it's on the opposite side of the relaxase, which is typically on the lagging strand, the one that enters the cell last. So since we know it's next to the ORT and the relaxase, we know it's on the leading region of the plasmid, which is great, because it makes us think that, well, it makes a lot of sense, right? If I'm invading a new territory, maybe the first thing I want to do is lower all the defenses, and then I can be more safe because I don't have a CRISPR and restriction and everything. And I apologize for the photographer for all the sport. Okay, that sounds great, but how general is it? So what I'll show you in this plot is the location where we found this anti-defense gene, where 0 is the first gene being transferred, and this is the more, the upper we go with, the higher we go with this value is following genes, and the minuses are the lagging strand. So this will be the last gene being transferred. And we checked anti-CRISPR, as I say, anti-restriction, anti-SOS, and this is the figure that comes out. And when I saw that, I was amazed, because I'm working on biological signal not for that long, but quite long, and rarely have I seen such a signal at first hand. So me and Burya were very happy, but then we thought, what if only one of these classes really produce the signal? Meaning, it's not that we see something that has to do with anti-defense. For example, just anti-SOS, which have been reported in a few cases to be in the leading strand, is what gives us a signal, and all the rest is just the noise. So we did it separately for anti-SOS, anti-restriction, and anti-CRISPR, and we see again and again and again this exact signal. You can see that the anti-CRISPR is more noisy. It's mostly because we don't know that many anti-CRISPR yet, and I'm sure it will become even cleaner moving on. Okay, so that was great, but we wanted more. So far, we looked on all the plasmids that were annotated as plasmid in NCBI. So basically, every researcher that says this is a plasmid, we said, okay, let's check it. The thing is that many researchers study bacteria. Many researchers study pathogens, not all bacteria, and specifically model organisms. So you have a lot of E. coli there. So we said we want to see how global it is. So we took all the genomic and metagenomic assemblies we could put our hands on, and then we looked for all those that have a relaxase and an ORT, so we have 206,000 sequences like that, and then we dereplicated them because we didn't want to see the same sequences again and again, and overall, on this 21,000 system, we measured the same measurement, and we saw again this very, 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 very strong peak. So we were convinced that indeed what we see is that the leading region is disproportionately enriched with anti-defense mechanisms. Now we want, before now, I want to show you two examples. So for example, here you can see the ORT there, and here's the triome, which is part of the relaxosome, and we can see we have an anti-CRISPR, another anti-CRISPR, anti-SOS, and anti-restriction, and a methyltransferase. This is one example from Salmonella, I think. Yes, Salmonella. And this is another one. This is a gram-positive bacteria, and you can see the same pattern of islands enriched with anti-defense genes. what else is there? So we know that these islands have a lot of anti-defense genes. There might be other interesting stuff in there. So what we did, we took the biggest gene families in this island. They appear here. So this is just the 100 biggest gene families that were enriched in the islands. And one thing we find, of course, is what we were looking for. So anti-SOS, anti-CRISPR, and anti-restriction genes. I should say that some of these genes, for example, the highest bar on the right, were specifically that one was 99.6% unknown genes, but 0.4% was a putative anti-restriction that is well known. So I think this might very well represent a distant homology of a known anti-restriction, and it would be interesting to see if indeed it has anti-restriction properties. What else did we see? We see a lot of things. We call them anti-defense related. So for example, I'll give one example that I'll give quickly is the DNA methyltransferases. These are not known to be in any way associated with restriction modification system, but the way restriction system work is to identify a methylated site and not cut them, and to do cut unmethylated site, meaning methylation is used in order to differentiate self from other. So the genome methylates its own gene, not to cut itself. And it's easy to imagine how a plasmid will have a DNA methyltransferase in order to methyl his own genome, and then it won't be restricted. Additionally, we also saw, of course, other genes marked here in grayish-blue and uncharacterized genes. Okay, it's all nice and neat, but I've mentioned that it's only one strand that is passing. So this gives us a few problems. The first problem is we need to make sure that these are on the right strand, right? Because if they are on the opposite strand, it doesn't help me that they are on the leading strand, because until the DNA won't be double-stranded, it won't be able to express it. So I'll mark here the ones that are on the wrong strand. They cannot be expressed. And you can see that 100% of the cases in the largest family are either known not to be related to defense or are unknown genes, which is encouraging. But still, there are people here that know cell biology much better than I do, but we do know that single-strand DNA is not what RNA polymerases use, right? It uses double-strand DNA. So we had a nice story, we had a nice peak, but if it cannot express from single-strand DNA, our story is not worth anything. And we were bummed, but just very briefly, because we found a paper from two researchers from Tokyo University back in the end of the 90s that showed in vitro that sequences within F plasmid can create a double-strand-like structure. So this is a single-strand DNA. It starts over there, and then it goes and creates a secondary structure that looks like double-strand DNA, and that's enough for the RNA polymerase to identify it and create a transcript in vitro. So that was great. Since we have AlphaFold 3, we did the exact same thing we tried to recapitulate what they did in vitro in silico, in the computer. What you see here is a RNA polymerase holoenzyme. And the thing here that looks really like double-stranded RNA within the RNA polymerase is in fact a single-strand RNA. You can see here, this is where he loops. So you can actually, this is a single-strand RNA. DNA, sorry. Single-strand DNA inside the RNA polymerase, and you can see this is the loop. So it's really, it looks double-stranded, right? Great. Do we have those in our leading region? So we searched for this kind of construct in the leading region. We found thousands of them. We looked for similar but not identical constructs and found thousands more. And now we are in the position where we know where they are. We have an hypothesis how they express, and the reviewers want to see proof. So we work just, like a year and a half and provide them some proof. It was pretty hard for us because every experiment we did here was the first time we performed that experiment, but we managed to show the following. So what we did is we said, okay, let's do a synthetic attack on the plasmid that I know exactly what attacked the plasmid and and how to defend myself, because part of the problem is that we don't always know which gene defends from which systems. So we said, okay, we'll take Cas9 with a guide RNA that target the plasmid. And if it works well, it would express the Cas9, and then will attack the plasmid, and there is no plasmid. So if I put a selective marker on the plasmid, like antibiotic resistance, it won't be able to grow because there's no antibiotic resistance, because the CRISPR destroyed and cleaved the plasmid. But according to our theory, if I put anti-CRISPR in the right orientation with the right regulation in the leading region, it will express the anti-CRISPR, it will block the Cas9, and then hopefully the plasmid will stay. We obviously need control. So first control, let's put it on the other strand, where I don't expect it to be very effective. Let's put it on the lagging strand, and that also is not supposed to be very effective. Okay? We're good with the experiment? Great. So, to the results. So first of all, what if I don't have an anti-CRISPR at all? So we did it in two settings. In one of them, we have a guide RNA that attacked the plasmid, and in that case, you can see the plasmid, the bacteria can't grow, meaning the plasmid was destroyed, there was no efficient conjugation. And as a control, we check the exact same system with a guide RNA that doesn't attack the plasmid, so you can see the difference between attack and no attack. Now, when we put the anti-CRISPR on the leading region, this is what we see. So we see basically it's almost as efficient as the Cas9 was never there. If we put it on the lagging strand, on the inverted strand of the leading region, we can see that there are some effects. So at some point, the anti-CRISPR can overcome the Cas9, but orders of magnitude less. This is a logarithmic scale. So we were happy. Gladly, the reviewers were happy, and we could summarize our project saying that we think the leading region include an array of anti-defense genes that are intended to protect the plasmid from all kinds of defense mechanisms, CRISPR, SOS, nucleases, toxin, and all those new ones that are being discovered all the time might also have anti-defense on the plasmid. And the schematic representation of plasmid in many papers, which every time I saw it made me very happy, was, well, there's the replication part of the plasmid, there's the adaptation part, that's where the resistance genes will be, silent pandemic, we are in a pandemic session. We have the propagation, this is all the system that creates the conjugation itself, but the leading region is always described as plagued with genes of unknown function. It described that it's used for stability and establishment, and now we claim that a major part of this stability and establishment are anti-defense genes. But I still have close to 10 minutes, so I'll be happy to tell you what we are working on next. So one thing is to identify a new system. This is something that Chen and Shachar, Chen is a master student, Shachar is a project student working in the lab. And basically, we say that we know where to find out as the defense genes. Let's see if we can actually find them. I should say that's what we feel pushed by the community to do, so we are starting to look into it. And what you can see here is structure of something we think is a putative anti-CRISPR that was unannotated but found in the leading region. And you can see that the structural similarity is striking. And we have another anti-CRISPR. But these are relatively easy to find because nowadays looking for similar structure on large database is becoming exceedingly easy. Another thing we are doing is trying to see whether we can find genes that bind to defense systems. So in this case, this is a restriction enzyme, and this is a known anti-restriction. And we can predict the binding of the restriction to the anti-restriction enzymes. And the question is, can we search for other proteins that bind similarly or to same spot into a restriction and other defense mechanism? I should say that I never thought I'd be a protein biologist, but these things are becoming so easy that we have to look into them. Another project, this is led by obviously somebody that's called Ori, is to find new orities. His last name doesn't start with a T, but I'm working on it. So remember I told you we had a ton of, we have 44.9 million genome and metagenomic region, and I said, contigs, and we had 200 and something that had relaxases and ORT. What didn't I tell you? I didn't tell you that about 1 million had relaxase, but as you can see, only 20% of those had relaxase and origin of transfer. Now, this doesn't make a lot of sense because the relaxase is the big protein that makes all, as I mentioned, it creates the nick, it transfers the plasmid. So there's not a lot of sense of encoding for a relaxase without having an ORT, something the relaxase to bind to. I'm sure we can think of scenarios like that, but we believe that, at least in the majority of cases, we are simply unable to identify the ORT. So Ori, being a bioinformatician, what he does, it looks into a lot of different features of the DNA at this region. So we look at secondary structure, at the genomic context, because we know now it will be next to anti-defense genes, and other physiochemical property of the DNA in that region, and he manages to predict pretty well. I don't know if you can see that, but basically he managed to predict pretty well, seems to predict new ORT. I'm saying it seems to predict new ORT because we haven't tested them yet, so this is still before the part that we start to work hard in order to prove it. And the last thing I want to talk about, which is the one I'm excited most, is all those single DNA promoter, the single-strand DNA promoter. From all the story, this is what excited me the most because I had no idea these exist. And now in a joint project of Chen with Buria and Karin from my lab, we are looking more deeply into it. So one thing we do is sequence similarity searches, which is what we did, basically smart blast or not so smart. The other thing is to look into the secondary structure because obviously what is important here is not necessarily the sequence, but the fact that it creates a double-stranded structure, and this didn't give us enough results for us to be happy, so we also use the machine learning that again takes similar properties and try to learn more and more patterns that identify these secondary structures, the single-strand DNA that looks like single-stranded DNA that looks like double-stranded DNA. I'm becoming tired, sorry. The other thing we want to do is look the other way around. So here we start to look at some things that have the same structure or same patterns as known single-strand DNA promoter. The other way is just to look at all the promoters in the leading region, cluster them together, and say, can we say something about them? Do they match those? Are they different? So we can see here an example for a few of our top clusters, and you can see, and here are their structures. So you can see that this one, for example, was not identified by BLAST, but was identified by the machine learning. Here we have ones that were identified both by the secondary structure and the BLAST, and some that were identified just by the secondary structure. So we are able to find additional clusters of promoters. You can see there how they look like, and some of them look like they could really be double-stranded DNA imitating promoters. Others don't look like anything to us, but it will still be interesting to study them and look into their function. And what we're doing now is trying to reconstitute the in vitro assays that we saw, which are they are pretty simple. I'm saying that, but we are still working on it, because basically what we want to do is put the single-strand DNA, our RNA polymerase, a few more magic ingredients, and we can check if we have the transcript or don't have a transcript. So this is something we're currently working on. That's it. I want to thank, again, first of all, I want to thank Bruria, Karin, Ori, Chen, Shirley, Maya, and Shahar, which all of them took some part in the things I've showed you today. I want to thank my group here in Up Here Times, and I'll be happy to take any question you might have.